We are in a world where we are searching for innovative and sustainable solutions. Precision fermentation can play a very important role. It's a groundbreaking role, actually. It can produce proteins or even tailor-made ingredients. At Wageningen Food Biobased Research, we are active in this field and we are searching for solutions to scale the technology. There are urgent needs from industry to get there. Today I'm here together with my colleagues and we will talk about that. Martijn, what is your view about the scalability of the process?
Thank you for your question, Joost, and that's an excellent question. Let me explain to you a little bit about scaling. So, in a nutshell, we can scale these type of processes. However, there are significant challenges as industrial scaling isn't a linear process. Let me break this down into two brackets for you. Firstly, it is essential that we understand all the parameters that affect the industrial scale process. Local conditions like oxygen concentration, temperature and pressure may vary within the system. We address this by using two types of approaches. Firstly, we use high throughput screening technology to understand how the local environmental parameters like oxygen concentration, temperature and nutrient concentration affect the process. Then we combine that information with advanced real-time monitoring tools to continuously steer the process as it is running at an industrial scale towards an optimal process. Secondly, cost reduction is a key factor for most of our industrial partners. Moving from batch type of fermentation to continuous type of fermentation represents a key opportunity for cost reduction. However, this comes with its own specific challenges, like buildup of mutations in the production strains and contamination issues. We are currently developing a body of knowledge on how to manage these issues to ensure that we can predict and prevent system failures.
Wow. Thank you so much. So I hear clearly you are very optimistic, correct?
That's definitely correct. We can assist many companies still with optimizing their process to further do further cost reduction and assist them with all their scale-ups.
So when we have the right scientific insights, you are confident that we can come up with a process which is scaled to an industrial relevant scale?
Definitely, and we've shown that before that we can definitely assist our clients and industrial partners with scaling their process in a reproducible manner.
Great, thank you so much.
You're welcome, Joost.
Next, I would like to introduce Sanne. Sanne, we heard from Martijn that scaling is feasible, but we also know that a lot of initiatives fail because of the high development cost. Can you elaborate what the success factors are?
Yes, of course. Thank you, Joost. Having a nice new idea for a protein or a product is fantastic, of course, but in order for a process to make it at scale, it needs to be cost effective from the start. So we do not only need to think about fermentation, but also about what comes after. For fermentation, there are three key points to focus on: titer, rate, and yield. Or in other words, how much are we producing, how fast are we producing it, and how efficiently are we converting our inputs into the product. And all of these three will impact overall process economics, and it's hard upfront to determine which one has the most impact. So it's important to early on investigate that. Then after fermentation, the protein needs to be purified from the final fermentation slurry before it can be used in a food formulation. And for this downstream processing, the rule is the more complex, the more costly. So factors that impact that complexity are the titer at the end of your fermentation. More protein means easier purification, but also the degree of purity that's required for your protein has an impact. Small contaminants can impact off flavor or maybe even functionality of your protein. For success, it's key that you latch on to these aspects early on in the development of your process. So early on you start testing, how do titer, rate and yield affect my process? What do I need to do for downstream processing? And at WFBR, we can help you with that. We have various platforms for testing, fermentation, optimizing these parameters early on in the process, so we can help you reduce risk later on.
That's very impressive, Sanne. Thank you so much. So if I understand correctly, when companies are setting up these processes, they really have to involve us as soon as possible, very early on.
Yes, early and then with the end in mind.
And with the end in mind, you mean the product? Because the proteins needs to be included in the product in the end.
Yes, definitely.
So they require functionality. And therefore, we have our experts, Laurice Pouvreau, who can tell much about protein functionality.
Thanks, Joost. Performance in food application is the key to success for any new type of ingredient. And it's no different to precision fermentation protein. Best practice is to be able to assess this functionality or its behavior as early as possible in the process, having in mind the end product quality you are aiming for. The first challenges with precision fermentation protein is actually the scale or the amount of samples we have available to assess their performance. Working or collaborating with an academic partner can be helpful. We have methodology to be able to assess the functionality of the ingredient at small scale. And we have the expertise to translate this result into real application. The second challenge of precision fermentation protein is flavor, meaning taste and aroma perception. The European rules really offer us challenges in having reliable data early enough in the process to assess these issues. By combining state-of-the-art analysis and sniffing or smelling experiment, we can assess and identify these challenges as early as possible in your process. The nutritional quality and the bioactivity of your ingredient is crucial for its success. We can assess at small scales by using a broad range of bioassays this factor. Let me give you an example. We can test at very small scale if the protein is digested in similar ways as the animal counterpart. We can also evaluate if inflammatory factors are present. In conclusion, by focusing on this key area, we can help our partner focus on the most promising development and bring precision fermentation protein from lab to table as quickly as possible.
That's a great conclusion, Laurice. Thank you so much. It's all about functionality, and I think we have a lot to offer to our partners out of the industry. I think this talk really makes it clear. I would like to thank you all, and let's have a coffee.
Super.